Hi there, my name is Janet Lorv, and I would like to briefly present to you my work on the identification of a putative inter-phylogroup horizontal gene transfer of the type 3 secretion system genes between distantly related Pseudomonas syringae strains. So to start off, just to provide you a bit of background, Pseudomonas syringae is a ubiquitous phytopathogen found all over the world. It is a very diverse species complex that contains both pathogenic and non-pathogenic variants, and because of this high genetic variability, it can actually be split into 13 phylogroups, under which some strains have actually unique species classification. But the phylogroups that I'll be focusing on is actually a subphylogroup 2C, where there's at least one isolate that is known to be non-pathogenic and have been isolated from healthy plant leaves. But the interesting part is that it contains this atypical variant of the type 3 secretion system, which is actually a key determinant of virulence in this species. In fact, based on other studies, we actually know that this secretion system variant is actually active and can actually inject effector proteins into its host plant. So the objective of this work is actually to analyze these group 2C strains and their type 3 secretion system and compare it to other strains in this species complex. And to kind of do so, we will be using the complete genomes that we previously assembled for two putatively non-pathogenic group 2C strains, which are called Pico 508 and Pico TLP2. So if I could just bring your attention to our methods, which is on the bottom left, what we did was cluster our two complete genomes with a selection of 416 Pseudomonas syringae reference strains, as well as 10 diverse Pseudomonas strains that will be acting as an outgroup. So in total, all 428 genomes were clustered together with Pirate, and due to the high genetic variability, we clustered them at progressively increasing identity thresholds starting from 50% to 95%. And out of all the gene families that we found, we were able to detect a hardcore genome consisting of 2,087 gene families that are single copy alleles in all strains. These gene families were then aligned together with MAFT and then passed to FastTree to generate a species tree from the core genome. In parallel, we also identified the pathogenicity islands that encode the type 3 secretion system via the HRC gene families found in all strains, and the DNA sequences of these genes were extracted and then aligned with MAFT, and a maximum likelihood tree was generated using RaxML with 1,500 bootstrap trees for support. When both trees are actually compared, as you can see in figure 1, which is the top figure under results, we can see that there is incongruency in the species placement between the two trees. On the left is actually the core genome phylogeny, where, as expected, the Pseudomonas syringae strains group together into their respective phylogroups, and the 10 outgroup strains actually distinctly cluster outside of this species complex, as expected. From this tree, we can actually confirm that our two sequence strains belong to group 2C, as shown in the inset and are highlighted in black, and also that this subgroup is actually larger than previously reported, where it actually now contains the strains that previously belonged to Pseudomonas fluorescens. And when we compared the gene families shared with its neighboring pathogenic phylogroups, namely phylogroups 2, 3, and 6, we were able to determine that less than 50 gene families are unique to group 2C, and they were basically primarily belonging to the atypical type 3 secretion system. On the other hand, the tree on the right is the conserved type 3 secretion system maximum likelihood tree. The tree topology indicates that all three variants are distinct from one another, grouping into three separate groups. And despite the fact that both the canonical and the atypical variants share the same tripartite arrangement, which you can see in figure 2, there is actually low sequence similarity between the two. And when we actually looked at the same gene families, we were actually able to identify that a subset of group 13 strains also contain this atypical variant, albeit excluding the flanking effector loci that are found in the tripartite variants. In fact, in our multi-loci analysis show that the two distinctly related phylogroups actually group together in this maximum likelihood tree, as seen highlighted in the red boxes. And we suspect that this incongruency between the core genome phylogeny and the ML tree indicates that the most common recent ancestor of these groups acquired the type 3 secretion system loci through horizontal gene transfer rather than vertical inheritance. And this is not the only instance of a possible inter-phylogroup transfer. As you can see, the same type of incongruence in the species placement between the two trees when you look at phylogroups 2, 3, and 5, where the difference suggests that phylogroup 5 actually gained the same variant as 2 and 3 from these other groups. We also analyzed the genetic structure of the secretion system variants by observing the gene arrangement of the pathogenicity islands that encode them. This is summarized in figure 2, which is actually the bottom figure of the results section. And from this, we can see that the atypical variant seems to have more of a hybrid characteristics between the two other variants. And despite the fact that it has a tripartite arrangement where usually a core loci is flanked by an effector loci, the core loci of the atypical variant actually shows more similar characteristics to the single partite variant than the canonical tripartite variant. And this is especially evident when you look at where the highly conserved gene AVRE1, it is actually found in the hypervariable region similar to the single partite, while the HOP-M1 gene is found in the effector loci, which is more common to the canonical tripartite variant. So kind of with this hybrid atypical variant in mind and its characteristics, we suspect that the possible recombination of only the core loci may have occurred between phylogroups 2C and 13. Whether this recombination was direct or indirect, like say through intermediate strains, is unclear at the moment, but when we take everything all together in this work, we would like to conclude that our analysis seems to suggest that the distantly related phylogroups 2C and 13 share an atypical type 3 secretion system, possibly through inter-phylogroup horizontal gene transfer. And just to wrap things up, I would like to thank you for listening, and if there's any questions, feel free to contact me through email. Thank you again.